We first started working with MDC in 2018 and were able to come together to create a highly successful grant which is now completed. And that really helped us to extend our wet lab capabilities and perform experiments that otherwise wouldn't have been possible.
When Andy first came to us in 2018, it was 4 people with new ideas to apply single molecule imaging. The catalyst for us was a successful Innovate UK funding award. That collaboration was an opportunity for SMi to build their first prototype device and install it in the MDC labs. That project was all about demonstrating early proof of concepts for detection of infectious disease, building and testing prototypes both in SMi's hardware and in MDC developing prototype assays.
SMi launched in 2021 as a team of 4 and has now grown to 18 people working across our UK head office and our US laboratories. SMi was founded because I had my own research laboratory at Cambridge University, and we had challenges measuring the sorts of experimental data that was needed for drug discovery. I had a colleague in the States who was an optics specialist, and I set him the challenge of developing a new microscope that could look at the single molecule interactions that are the keystones of drug development. He provided us with an instrument in a very short space of time, and we were able to take that instrument into those that we have today. Those instruments are now providing us with unprecedented data and allowing us to see the key interactions that govern biomolecular interactions and to allow us to develop drugs and new assays for diagnosis. At SMi, our vision is to advance biomedical research and diagnostic testing by making previously complex experimentation and analysis into something that's simplified and can be used regardless of your level of experience. Collaborating with Medicines Discovery Catapult has enabled SMi to accelerate our research and leverage MDC's expertise to explore new market opportunities.
In our current project, we're applying SMi's single molecule approach to the detection of circulating tumour DNA as well as protein markers. We want to show what a diagnostic test could look like and work out how accurate and sensitive it can be compared to other technologies. MDC has been able to support SMi in lots of ways. Applications for funding, sharing of expertise in assay development and validation, access to benchmarking technologies, and even access to clinical samples.
My name is Andrzej Rutkowski. I'm a lead scientist in biomarkers at Medicines Discovery Catapult. Previously, I worked with liquid biopsies and digital PCR, both of which are quite relevant to our collaboration with SMi. So liquid biopsies are a very powerful tool when it comes to cancer diagnostics and cancer research. Liquid biopsies are biopsies that do not require to cut open the patient. You can just collect some blood from the patient and analyze what's in that blood, and that can give you a surprising amount of information about the state of the patient. When we look at something called circulating tumor DNA, we can detect mutations that drive the cancer. And depending on what mutations we discover and how many of these mutations we discover, we can, for example, say what the trajectory of the cancer will be, how quickly it's going to progress, what the prognosis for the patient will be. We can also say whether this patient will respond to the specific treatment. And just as importantly, when the patient has been treated for cancer and we believe that the cancer has gone, we still need to monitor the patients to make sure that the cancer doesn't come back. And liquid biopsies have the power to detect a relapse of cancer months before such technologies as imaging. Here at MDC, we can provide access to cutting-edge technologies for analysis of liquid biopsies, such as digital PCR. It's a technique that can detect single molecules of DNA, including the mutations, the cancer-driving mutations that I talked about. And this is currently the gold standard in analyzing liquid biopsies for cancer DNA. With our access to these technologies, we'll be able to benchmark the SMi platform against them to make sure that we help them develop as sensitive method as possible.
So I'm Regine Anderson. I am a senior scientist in the biomarkers team here at Medicines Discovery Catapult. My main responsibility is to source human tissue samples for the various projects that we're involved in. So that's my main focus, and that encompasses everything from procurement of those samples, doing feasibility studies, managing the ethics, and once the samples are on site, managing the life cycle of those samples. Because of our existing relationships, it was easy for us to know where to go when sourcing the samples for this project. We have put a lot of focus into building relationships with various tissue providers, and that spans across different types of biobanks. So we're well equipped to source samples based on the collaborators' needs. When this project came about with SMi, it was very clear which biobanks would be more suited because of those relationships that we've developed. We're often informed about new cohorts that are starting and different collections that biobanks have, so it was very easy for us to get started with this project. One benefit of the provider that we chose for this project is that they span across 5 different hospitals across the north of England with a range of patient demographics. So because of that, we now have confidence that the samples that we'll receive will be quite varied, and hopefully that will limit any confounding factors when analysing the data. So we're able to overcome some of the barriers of drug discovery, one of those being cost. As part of the network that we've developed with different sample providers, we have a select number of providers that offer samples at a very affordable rate. They focus mainly on cost recovery as opposed to revenue generation.
My name is Naomi Gibson. I am an assay development research scientist here at MDC. And this project with SMi is a really exciting opportunity to be able to work with a technology at the cutting edge, early stages of development. As an early careers researcher, it's quite a unique opportunity to be able to work with technology at this early stage and with prototype instruments.
We work closely with their teams in the UK and the USA, and we're constantly feeding back our evaluation data and our ideas. I think this approach really helps to move the project forward and to build a strong relationship with the company that we're working with. Plus, we have many different areas of expertise and we're able to be thorough and look at a project from many different angles. What's exciting for me about this project is to see the potential of a new company such as SMi, their science and their people, and then to have this shared goal and feel that we've really had an impact on that part of their journey. I wish them every success. They're a great company.
Matt Burnham working on the project has been tremendously helpful because although we've had 2 separate Innovate UK awards, Matt's consistency across both of those awards has allowed us to hit the ground running and has allowed us to ensure that our products are suitable for our ultimate end customers. Through Matt's support with his team, we've been able to take an external validation of our technology and to ensure that we're not looking just from an internal perspective on what we're developing, but that it's suitable for a much broader range of scientific uses from people with a broader range of experiences.